బాగుండు బాగా వచ్చింది బాగా బాగా వేరు బాగా
Hello, students, and welcome to another session for Basics in Biology NPTEL course. Today, we'll be following lecture week 7, in which we'll be solving example questions from lecture week 7, right? So, it's already 3:05, and I'll keep people who are requesting to join this call, I'll keep admitting them, while this will be a recorded lecture as well as live session where the recordings would be available on YouTube. So I will teach in a manner in which the people who are watching this recorded video in YouTube will also benefit from this class. Okay, so quick bit of an introduction. My name is Ashutosh Behra, and I'm a PhD PMRF scholar from IIT Kanpur. And me, along with uh, other PhD students, are teaching assistants for this course. This course is Basics in Biology, NOC BT 25. This course is taught by Professor Vishal Trivedi at IIT Guwahati. So today we'll be dealing with lecture week 7. So we have already solved these sessions from starting from week 1 to week 6 in the past 6 weeks, and the slides as well as the recorded lectures are uploaded through the NPTEL, and they have been provided to you. So this is the order of the session that I'm conducting here. So we will have a set of 10 to 15 questions which are examples from similar kind of questions which you will face in your assignments, in your quizzes as well as in your exams. These will be multiple choice questions, and we will follow them up one by one, clicking at and gaining correct answers from the students, the audience, and then I'll reveal what is the correct option for this question, and then we will understand the concepts and the logic behind. We'll cover the topics behind these question and answers. Okay, without any further delay, yeah, without any further delay, I'll share my screen with everyone. These are the general instructions for all the students present in the live session. I would request everyone to please turn off your mic and video chat for the smooth conduction of this session. Uh, this session will be an interactive session where you can interact with me via the chat box. Right, so I would request everyone who is joining this session to please turn off your mic and video chat. Keep it turned off during this session and interact with me and answer the questions via the chat box. I'll be following up with your answers and questions through the chat box with a mirror here. I've joined here. So as students are requesting to join this call, I'll keep admitting them, and let me share my screen. I hope my video is I'm audible. Someone has raised a hand. Okay. Bhagya, do you have any questions? You can speak up. Bhagya. I think Bhagya has a question. Okay, so guys, if you have any questions, you can post it in the comment box. And for the smooth conduction of this session, I would uh, I would request everyone to use the chat box and turn off your video chat and your mics. Okay, let me share my screen with all of you. Okay, I would ask whether the students can see my slides, and if my voice is clear and audible, then we will start with this session. You can let me know in the chat box with a yes, or you can like. I'll follow it up. Can you see the slides changing, and is my audio clear and audible? Students, if you please respond, I would know. Just type a yes or no in the chat box. I would know. Okay, great. So once again, I welcome everyone to lecture week 7 for Basics of Biology. The instructor for this course is Professor Vishal, and my name is Ashutosh. I am the teaching assistant for this course. And if you remember all the lectures taught in week 7, the questions will be from those lectures. These will not be the exact assignment questions that you will receive, but exemplar questions from the same topics. So these questions may or may not be similar to what you face in your quizzes and your assignments, but solving these questions will help you understand the concepts better and also enhance your reasoning and ability, memory skills. Okay, so this is the path of this is the path of the or the flow of the class that we will follow. I will post a question on the presentation on the chat on the screen with four options, and out of which one of them is correct, multiple choice options, right? And I will ask the chat and the live participants as well as the students who will be watching this video to solve the answer. I'll wait for a minute and then collect all the answers that have been answered in the live session, and then reveal the correct answer, and then we'll briefly understand the concepts behind the correct answer. Okay, so the first question of this session is here. Question number 1: the antigen binding site in an antibody is composed of dashed. The antigen binding site: option A, variable heavy chains only; option B, variable light chains only; option C, constant heavy and variable light chains; option D, variable heavy and variable light chains. So this is the first question. These are the four options that is posted here. Now I want you guys to tell me what is the correct answer. You do not need to shy off. It's okay if your answer is wrong. Even if you are doing guesswork, we will come to know what is the correct answer and then also understand the concepts behind it, right? So I would encourage all the participants in the live session to post your answers in the chat box. Let us wait for a minute to collect some answers in the chat box, and then I will reveal the correct answer to this question. Understood? Okay. I will read the question once again for you guys. The antigen binding site in an antibody is composed of dash. Option A, variable heavy chains only. Option B, variable light chains only. Option C, constant heavy and variable light chains. Option D, variable heavy and variable light chains. After a minute or so, I'll check the chat box if people have answered. You can just mention the options, whatever option you think correct is in the chat box. Meanwhile, students who are requesting to join this call, I will be admitting them. And for all students who have joined recently, please turn off your video chat and mic for smooth conduction of this session. You can interact with me via the chat box. Here is a question posted for you guys. The antigen binding site in an antibody is composed of dash. And you have to let me know in the chat box with the correct option. Okay, it's over a minute. Time for me to check what students have answered in the chat box. Oh dear, I don't see any answers in the chat box. Okay, maybe students have did not have the time to go through these lessons. Okay, but from the next question, I would expect at least some few answers. We are 4 to 5 participants here, as well as those who will be watching this video in the future. Please try to answer by yourself. This is for a dialogue between you and me, as well as me to judge, me to know what kind of understanding you have of this concept, right? It will also help brush up your memory for these lectures and help you in learning the concept as well as attending your exams. So, time to reveal the correct answer to this question. The antigen binding site in an antibody is composed of variable heavy and variable light chains. Option D is the correct answer. The antigen binding site has both variable heavy and light chains. So let us look at the structure of an antibody. If you remember, the structure of an antibody in general is a Y-shaped structure. This is the structure of the antibody. It is a Y-shaped structure, and in the two arms of these Y are the antigen binding region, or which is known as FAB. Fab regions or antigen binding regions, where AB stands for antigen binding. And then the stalk of the Y is called constant region or FC region, which is also known for its biological activity mediation. So these two arms of the antibody will bind the antigen, while this arm of the antibody will mediate its biological activity. So if you look closely at the antigen binding region or the FAB region, it is consisted of a constant chain a big constant change, long constant chain. By chain I mean a chain of polypeptides, and a heavy chain and a light chain, which is here demarcated in two different shades of green. The light chain is depicted L, while heavy chain is H. So an FAB region is constituted by variable heavy chain and the variable light chain region, while this region is the constant region, constant heavy chain, constant light region. So heavy chain hyper variable regions. So these are hyper variable regions which can undergo recombination to form different combinations of different antigen binding regions, so that only within a minor changes of the coding region of an antibody it can recognize a totally different antigen. And here an antigen can be a pathogen or a self antigen as well. So these FAB regions are not only made of variable heavy chain but also variable light chains. Only the constant region is made up of heavy chains. So constant region is purely made up of heavy chains, while antigen binding FAB regions is made up of heavy chain light chain hybrids. And there are these hyper variable regions which with minute changes can identify a variety of antigens, which makes antibody very versatile, and each specific antibody binding to a particular antigen. So what is this antigen binding site? Let us understand it better. The antigen binding site in an antibody is called the paratope. So the site on the antibody which binds the antigen is called the paratope, while the site on the antigen which is bound by the antibody is called the epitope. So there is an interaction between an antibody and an antigen. A very specific interaction is between a paratope and an epitope. The antigen binding site in an antibody is called a paratope. It is highly specific region of the antibody that recognizes and binds to specific epitope on the antigen. This is what we just discussed. Now what are the features of the antigen binding site or the paratope? What is the feature of this paratope? Where is it found? It is found in the variable regions of the antibody heavy and light chains. So this is what was the answer to this question. Specifically within the complementarity determining regions. So even within the long heavy chain and light chains, there are regions which are called complementary determining regions or CDRs. These CDRs are highly variable and very specific to certain specific epitopes on an antigen. And what is the composition? Formed by 6 CDRs, 3 from the heavy chain and 3 from the light chain. CDR1, CDR2, CDR3 of VH, which is variable heavy chain region and variable light chain region. And what is its specificity? The structure and amino acid composition of the paratope determines its binding affinity and specificity for a particular antigen. So for any given antigen, only a certain combination of the heavy chain and the light chain, the structure and amino acid composition is very unique, and that forms the paratope region, which recognizes the epitope region on the antigen. And mostly these antigen antibody interactions are governed by non-covalent interaction, and the paratope can recognize a variety of epitopes including linear peptides and conformational epitopes. So there is structural diversity in a paratope. The paratope epitope interaction is non-covalent. It is located in the variable heavy and variable light chain regions of the FAB fragment of an antibody, right? Binding occurs through hydrogen bonds, van der Waals forces, electrostatic interactions and hydrophobic interactions. So this is the antigen antibody interaction, and the antigen binding site is constituted both by variable heavy chain and variable light chains. Now, is it clear to the students? Just let me know with a thumbs up or a yes. Very good. Good, great. Moving on to the next question, question number 2. Antibodies are secreted by dashed. Antibodies are secreted by dashed. Option A, macrophages. Option B, plasma cells. Option C, T cells. Option D, dendritic cells. Which cells primarily secrete antibody? The hint is given in the question as well, right? Antibodies are secreted molecules. And what kind of molecules? These are proteins. Which cells primarily secrete antibodies? There are four options. I think it should be easy. Macrophages, plasma cells, T cells, or dendritic cells. Let us wait for a minute for all the students present in the live class as well as people who are watching this video to answer. I will reveal the correct answer in a minute, and we will discuss further on this question. Someone has raised a hand, Manjulata. Guys, if you just have to answer, you just need to post your answer in the chat box. Whether you write the options or the terminologies, I will follow it up. If you have any questions outside or related to these questions that I am posting, then you can raise your hand and I may ask you to talk, right? I see people are actively responding. Very good. That's the spirit. So this class is an interactive session, and I want all of the live students to gain this advantage. Interact with me through the chat box. Answer these questions, and this will refresh your understanding and your memory of these lectures and help you solve your own assignments and quizzes. Which of these cells are primarily secreting antibodies?
Plasma cells.
Yeah, I think Shanmugha spoke up. Yeah, Shanmugha, you please post your answer in the chat box.
Sir, okay, sir.
Yeah. Thank you. So, general instructions to everyone. Please turn off your mic and video chat during this whole session and post your answers through the chat box. I think it's time for me to check the answers. Beautiful. Shanmugha has answered plasma cells. Manjulata, Vedshri, Prashanti have all answered plasma cells. And you guys cannot be any more correct. The answer is plasma cells. Correct answer. Now, what are these plasma cells and what are its function, right? So we have two kinds of lymphocytes. Now, lymphocytes are the immune cells of our body. These are white blood cells. We have two major kinds of lymphocytes, the T cells and the B cells. And they are named as such because T cell births and develops inside the thymus gland, which is present in our just below our throat. The thymus gland, sorry, not the throat, the in the chest, right? And B cells are born and develop in the bone marrow. So two types of lymphocytes. Of those, B cells are the one which have the major task of secreting antibodies. Now what happens to a B cell? So there is a naive B cell. Naive B cell means this B cell doesn't know anything. It's just out of its birth. Now when does it gets activated? Whenever it encounters an antigen, whenever there is a pathogen or a self antigen which it has to recognize, this cell gets activated. And also with the help of T cells. T cells are helper cells. T cells help activate B cells. So within these two stimulus which are now coming to the B cell, a naive B cell can become a memory B cell. Now memory B cell can then turn into a plasma blast. Now memory B cell, because memory B cell will keep a copy of the particular antibody required to attack or to bind the antigen within itself, and now it will turn into a plasma blast. So what is a plasma cell? A plasma cell is just a B cell which is actively secreting antibody. It is activated B cell, right? So what are these plasma cells? Plasma cells are specialized, terminally differentiated B cells that function as antibody secreting factories. Their only job is to secrete antibodies and then die off. And these are differentiated B cells. So we have a naive B cell getting stimulus from the antigen as well as from T helper cells, stimulated into a plasma cell which is now terminally differentiated. Naive B cells have antibody studded into its cell membrane, while a plasma B cell or a plasma cell have antibody which can be secreted out of the cell. So plasma cells are specialized terminally differentiated B cells that function as antibody secreting factories. Key features of plasma cells and antibody secretion. Naive B cells are activated upon encountering an antigen. They differentiate into plasma blasts, as we just saw in the previous image, then mature into plasma cells in response to signals from T helper cells and cytokines like IL-6. So many cytokines play a major role in stimulating B cells to differentiate into plasma cells. Now there is antibody production. Plasma cells continuously secrete antibodies which are identical to the B cell receptor of the original activated B cell. So plasma cell, what are the antibodies? These are nothing but the secretory version of the B cell receptors. B cell receptors are the proteins that are embedded onto the B cell, which recognize the antigen initially. Then upon signaling and getting stimulus from the helper T cells, now these cells turn into plasma cells. These receptor itself now become secretory and are secreted out. Each plasma cell secretes only one class of antibody, IgM, G, A, E, D. These are different types of antibody depending upon the constant region of the Fc region, which we encountered in the first question, depending on the class switching guided by cytokines. These antibodies circulate in the blood, lymph and tissues, binding to specific antigens to neutralize or mark them for destruction. So now the antibody can roam around in the blood, lymph and the tissue fluids to mark the antigens for destruction or neutralization, right? And what is the lifespan and function of these plasma cells? These plasma cells are short-lived because only when there is an antigen, there is stimulation of naive B cells into the plasma cells which are terminally differentiated and they apoptosize or die. They secrete antibody for a few days to weeks before dying. They die off immediately. They are not permanent. But there are some permanent forms of plasma cell. Long-lived plasma cells reside in the bone marrow and continuously produce antibodies for years, providing long-term immunity. So there are some long-lived plasma cells which are a cousin of the memory B cells which are present in the bone marrow, and they produce antibody forever for providing long-term immunity against the body, against the antigen for the body. And what is the structure and specialization? Plasma cells have an extensive rough endoplasmic reticulum and a prominent Golgi apparatus to support the massive production of the antibodies, up to thousands per second. You see how massive the production of antibody is. From a single cell, thousands of antibody molecules, thousands of protein molecules can come out per second. And plasma cells have an extensive rough endoplasmic reticulum. Why? Endoplasmic reticulum is the place, endoplasmic reticulum and the Golgi apparatus is the place where protein folding rearrangement happens to give the antibody its Y-shaped structure as well as specificity to its specific antigen. That is why these plasma cells have a very well defined and very large endoplasmic reticulum and Golgi bodies. So this is a summary about all the plasma cells, and I'm very happy that you guys have all answered it correct. Plasma cells are the major antibody secreting cells of our body. Is it clear to the students? Just let me know with a response in the chat. Very good, very good. Okay. Moving on to the next question. Question number 3. In hybridoma technology, B cells are fused with dash. In hybridoma technology, B cells are fused with dash. Option A, T cells. Option B, plasma cells. Option C, myeloma cells. Option D, mast cells. If you remember the nits and bits of hybridoma technology and for what it is used, B cells are fused with which kind of cells? Are they fused with T cells, with plasma cells, with myeloma cells or mast cells? Let us wait for a minute and collect some answers, and I will reveal the correct answer in a minute or so. Right? Okay, it's time for me to reveal what is the correct answer. Let me check if students have answered. Shanmuga has answered myeloma cells, Manjulata myeloma cells, Vedashri myeloma, and Shravani has also answered myeloma. And the correct answer to this question is myeloma cells. Very good, very good. You guys have answered very correctly and great participation as well. I know it's a thin population, but it's always good to see students who pay attention and participate in these one-on-one sessions, right? So in hybridoma technology, the B cells are fused with myeloma cells. Now, what is this hybridoma technology we are talking about? Here is a schematic showing all the steps involved in this hybridoma technology. Let me brief you about why is this technology invented and what is the purpose of this technology. Hybridoma technology is the technology to produce long-lived plasma cells for massive production of antibody. Right? For massive production of antibody. So, and these antibodies can be used in immunotherapy, can be used in research purposes, and is being used, vigorously and variously across generations. Just a minute. Yeah. So what happens in hybridoma technology? There is an animal into which the antigen is injected. So first we need a natural system to generate the antibody or the B cell for our particular antigen, and this antigen can be antigen of our interest. It might be a pathogenic antigen of, let's say, a rabies virus, to which we want to develop an antibody as a vaccine. So the antigen is injected into the animal, which is called the immunization, and this animal can be a mouse or a rabbit or a horse. Then against this antigen, the animal produces B cells in the animal's blood. The lymphocytes, B lymphocytes, get differentiated into activated B cells or plasma cells. But these plasma cells, as we just saw in the previous question, are very short-lived, and they will secrete antibodies for a few days or a week and then die off. So how to increase the longevity of these plasma cells? So these plasma cells are now fused to myeloma cells. Now myeloma cells are a kind of cancerous cells. Myeloma cells are cancerous cells. So B cells are fused with these cancerous cells to give them properties which will let them divide indefinitely. And with fusion by PEG or electrofusion, so in this stage, the B cells can be fused to myeloma cells to produce a heterokaryon. And then we have something which is known as hybridoma cells. Hybridoma because it is a hybrid between a B cell and a myeloma cell. Now we have a hybridoma cell with expressing receptors of the B cells. Now these hybridoma cells are selected via multiple steps using selection medium and antibiotic screening to screen for the particular antibody which is recognizing our cognate antigen. And these cells now have the capacity to indefinitely divide using the property of the cancer cells, and thus they will secrete antibody as long as we want them to. And these cells now are immortalized plasma cells, which are now known as hybridoma cells. So these hybridoma cells will continuously secrete these antibodies called monoclonal antibodies. So this whole technology is known as hybridoma technology. So hybridoma technology is a method used to produce monoclonal antibodies, identical antibodies derived from a single type of immune cell. So we want the antibody to recognize a particular antigen, and this technology developed by Georges Kohler and Caesar Milstein in 1975. So these were the scientists who discovered this technology for us. Involves the fusion of B lymphocytes with myeloma cells to create hybrid called hybridomas. Then these hybridomas can produce specific antibodies while being immortal, meaning they can continuously divide and generate antibodies indefinitely. Okay. So what are these steps in hybridoma technology? We just saw in the previous. I think there is a question in the chat box. So what is meant by HAT selection? So I'm coming to this. If you follow this slide, we will come to this in a minute, and we also have questions on HAT selection. So what are these steps in hybridoma technology? First step is immunization. A mouse or any other host is injected with a specific antigen to stimulate an immune response. And then we isolate the B lymphocytes from the spleen. The spleen is harvested to extract the B cells. So the spleen cells, spleen is an organ in the mouse body or in mammal body, which is the storehouse for all lymphocytes. So whenever there is a primary antigen infection or the host is infected, then lymphocytes gather inside the spleen and then travel to the lymph nodes via the spleen. The spleen is harvested to extract the B cells to produce the desired antibody, and then these B cells, the spleen cells, are fused with the myeloma cells. The B cells are fused with the myeloma cells, cancerous cells, using PEG to form hybridomas. So now we have these hybrid cells. Now we will select them, selection of hybridomas. Now which hybridoma really has the antibody of our interest, the antibody which recognizes our particular antigen. So to screen that, the mixture is cultured in HAT medium. The question that was asked in the chat box by, I think, Shanmuga. HAT stands for hypoxanthine, aminopterin and thymidine medium. So these are three components, these are three selection medium, and why we use this medium we will come in the follow-up questions, which selectively allows only fused hybridomas to survive. So what is the role of this medium, selection medium? It will only allow fused hybridomas to survive. So when we fuse these B cells with the myeloma cells to form a hybridoma cells, not 100% of the cells will fuse and form a hybridoma. So select unfused cells to select only pure spleen cells with myeloma cells, and from the mixture isolate the hybrid cells. We use this medium, hypoxanthine, aminopterin and thymidine medium, which will allow only the hybridoma cells to grow while kill all the other cells, the spleen cells and the myeloma cells. Then screening and cloning, subsequent screening and cloning to using ELISA, enzyme linked immunosorbent assay, to screen for the antibody that is important for us or that is recognizing the antigen, and then mass production of such monoclonal antibodies from the identified clone of the myeloma or the hybridoma. The selected hybridomas are cultured in large quantities or injected into mice to produce antibodies in ascitic fluid. So this is the whole technology of the hybridoma procedure, in which you first inject the antigen to the mouse or the host or the rabbit, and then you isolate spleen cells, isolate B lymphocytes from there, take the myeloma culture, fuse them using PEG, polyethylene glycol, to form hybridomas, select those hybridomas which are perfectly fused using the HAT medium, and then select the clones and the antibodies using ELISA, and then use them for mass production of monoclonal antibodies. Is it clear to the students? Just let me know in the chat box. If there are any questions, let me tell you, we have follow-up questions. More next, the following questions will be on various steps of the hybridoma technology where we'll discuss it in further detail. Okay, thank you for the response. Very good. Moving on to the fourth question. The blood from a person with O positive blood group will show agglutination with dash. So this is a trick question again. You have to read the question very carefully. The blood from a person with O positive blood group will show agglutination with dash. Option A, anti A. Option B, anti B. Option C, anti A and anti B. Option D, anti D. You have to carefully remember all the kinds of blood group and the kind of antigens that are present on these blood groups. And what is the blood group in mention in the question? The blood from a person with O positive blood group will show agglutination with which of these antibodies? Is it anti-A? Is it anti-B? Is it both anti-A and anti-B or anti-D? Let us wait for a minute and collect some answers. I will reveal the correct answer in a minute or so. The blood from a person with O positive blood group. Okay, time for me to reveal the correct answer. Let me check whether the students have answered correctly. Shanmuga says option C, Vedashri, Manjulata, and everyone is saying option C. Shravani says A and B. Okay, okay. Then I think I have to clear this concept for you guys once and for all, because the correct answer to this question is option D, anti-D. A blood from a person with O positive blood group will show agglutination with anti-D antibody. It is not C. I'm sorry guys, but this time your answer was not correct. Both anti-A and anti-B will be shown by a blood group from a person with AB blood group. A person with AB blood group will be showing agglutination with both anti-A and anti-B. But for a person with O positive blood group, the anti-D antibody will show agglutination. So let us understand this blood grouping system in detail. So what is this ABO blood grouping system? The ABO blood grouping system is based on the presence or absence of two antigens, just two antigens, antigen A and antigen B on RBCs, and the corresponding antibody anti-A and anti-B. So whenever there is a prefix anti, it means the antibody, not the antigen, right? In the plasma. And if you look closely, for the blood group O individuals with blood group O have neither antigen A nor antigen B on their RBCs, but they both possess both anti-A and anti-B antibodies in their plasma. So this statement can be confusing for you guys. The blood cell, if you imagine a blood group O blood cell, imagine an RBC. The blood cell does not have any antigen, no antigen. That is why it is blood group O. No antigen A, no antigen B, but it has antibodies against antigen A and anti-B. So if you take the plasma of their blood, it has both anti-A and anti-B. So this is where you guys got confused. But the O blood group blood cell as such does not have any antigen. Now what is this Rh blood group system, the positive negative system? The Rhd antigen is the most significant antigen in the Rh system. If an individual has the Rhd antigen, they are considered Rh positive, and if they lack it, they are considered Rh negative. So O positive, the question was a person with a blood group of O positive, right? And what are anti-D antibodies? Anti-D antibodies are produced when an Rhd negative individual is exposed to the Rhd antigen. So now we have a person who is O positive. So the positive means it is expressing the Rh antigen. So the Rh antigen will be recognized by the anti-D antibody. Typically through pregnancy with an Rhd positive fetus or blood transfusions, we all know this condition where the fetus, which is Rh positive, and the mother, when it is Rh negative, can have complications when the antibodies from anti-D antibodies from the mother can try to kill the fetus. So on my left is a chart showing blood group testing. O positive will only show agglutination. Agglutination means aggregation with anti-D antibodies, not with anti-A, not with anti-B, because it does not have the antigen A and antigen B. It only has, since it is O positive, it only has the D antigen or Rhd antigen, so it will show O positive, will only show agglutination for anti-D antibodies. While for AB antigens, AB blood group, it has both these antigen, anti-A, anti-B will show agglutination, as well as anti-D will show agglutination if it is AB positive. So it is the other way around. Blood group O does not have any antigen on it, so it will not be agglutinated by anti-A and anti-B, but since it is O positive, it has the D antigen on itself, it will be agglutinated by anti-D antibody. So here is a chart for easy representation. That is what I was showing you. These are the different kind of antigens present on the surface of RBCs. Blood group O individuals naturally possess anti-A and anti-B antibodies in their plasma but lack antigens on their red blood cells. This is what I was trying to tell you. So when these red blood cells will not be agglutinated by anti-A and B, right? This is where you guys got confused. Anti-D antibodies on the other hand are formed in response to exposure to the Rhd antigen and are not naturally present in blood group O individuals. Group A, B, AB, group O. So A has this kind of antigen which is recognized by anti-B. A has a B kind of antigen. B has A kind of antigen which is recognized by anti-A. Group AB has both kinds, which does not have any antibodies in the plasma. Right? A has A antigen, B has B antigen, AB. A and B antigens are both present on AB, and with group O there are no antigens. So there is a difference between antibodies in the plasma, which are complementary to the antigen present on the red blood cell itself. I'm sorry. And the antigens in the red blood cell present over the red blood cell. That is why the correct answer to this question would be anti-D antibody, since it is O positive, and O does not have any other antigens on its surface to be recognized by anti-A or anti-B. I hope it's clear to the student for once and for all. This topic can be a little confusing, but if you follow these rule of thumbs, you will be able to delineate which are the antibodies, which are the antigens that are expressing on a red blood cell, and the antibodies which can agglutinate against it, and antibodies which are present in the blood of the same plasma of the blood cell, like type A blood group. These are the antibodies in the plasma, since the same antibody will not recognize its own blood cells, whereas this blood cell will agglutinate with anti-A antibody. Is it clear to the students? If it is not, then I can repeat the topic. You just have to let me know, is it clear to the students? Okay, there is a question. Is that happens in all negative blood mothers while they in gestation period? Yes, the complications may arise, Shanmuga, because the Rh factor is a very potent antibody. Anti-D antibodies are secreted in the mother's blood and they make a way through the placenta. Antibodies are small molecules which can make their way through the umbilical cord and the placenta, since the fetus is connected to the blood. So many a times it may lead to abortions, which is also called erythroblastosis, if I'm not wrong. Okay, I just saw a single thumbs up. I don't know if students are clear about the concept of antigen and antibody and agglutination. If you want, if you want me to teach it again, I will. Okay, I don't think we will stall any more time here. Let's move on to the next question. As you go through the lectures once again, and even this recorded session will be available in the YouTube. These slides will be shared to you. You can follow it up, read it by yourself, then you'll have a clearer understanding of this problem. Moving on to the next question, question number 5. Medium used for selection of hybridoma cells is. What is the medium used for selection of hybridoma cells? Option A, LB medium. Option B, HAT medium. Option C, SDA medium. Option D, blood agar medium. Come on, guys. This is what we have already discussed in the previous concepts. So that is why I keep saying that these sessions will proceed very slowly. I will not rush you guys. Take your time. Tell me what you think is the correct answer, and we will spend a lot of time in understanding the concept behind these topics, so that you never ever encounter any problems when you are dealing with these topics, right? In your exam, in your for your learning and concepts. So we had just discussed the hybridoma cell technology. Now, what is the medium that was used for selection of hybridoma cells? Let me wait for a minute to collect some answers, then I will reveal the correct answer. And I know that you guys will answer it very correctly. I see people are still requesting to join. Uh, I'm sorry for all those who have joined late, but we are already on question 5. You can follow up on those hereafter. We'll be solving 15 such questions, so 10 more questions to go, and then we'll end this session. I will try to finish as many questions as possible within the stipulated time of 5 p.m. since we are going really slow. Okay, it's time for me to check the comment box and reveal what is the correct answer. HAT medium. Shanmuga says HAT medium. Manjulata option B. Shanmuga has typed something I don't understand. Shravani has answered answer B. Saich has answered B. Someone has raised a hand. Sophia, yeah. You can tell me what is the question you have. Sophia, you can turn on your mic and talk to me. Or you have a, if you're not comfortable, you can post it in the chat box. I'm anyways following the chat box, right? Okay, so moving forward with the question. Meanwhile, Sophia is posting her question or her query. Correct answer is option B, HAT medium. You guys have answered really correct. HED medium is a selection media for hybridoma cells. Now, what is HED medium? As we have already discussed, but let us go a little bit deeper inside the concepts. HED medium stands for three components: hypoxanthine, aminopterin, and thymidine. Hypoxanthine, aminopterin, thymidine medium. Now, what is this medium? Is a selective culture medium. Selective culture medium used in hybridoma technology to ensure survival of only successfully fused cells. Cells which have successfully fused, the B cells and the myeloma cells which have successfully fused, those fusion cells will only survive in this HET medium while eliminating the unfused cells. Now, what are these components? Hypoxanthine is a purine derivative that supports the salvage pathway of DNA synthesis. Now, for nucleotide synthesis and DNA synthesis, there is a salvage pathway. Hypoxanthine is a purine derivative. Now, what are the purines we have? We have adenine and guanosine are the purines, and the pyrimidines are thymine and cytidine. So, hypoxanthine is a purine derivative that supports this salvage pathway of DNA synthesis. Aminopterin, a folate metabolism inhibitor that blocks de novo synthesis of nucleotides. And thymidine, a pyrimidine derivative that supports DNA synthesis through the salvage pathway. So, two of these components, a purine and a pyrimidine, supporting salvage pathway of DNA synthesis, and aminopterin is a inhibitor that blocks de novo synthesis of nucleotides. Now, what is this importance? It eliminates unfused myeloma. Myeloma cells used in hybridoma formation are chosen to lack the enzyme HGPRT, hypoxanthine guanine phosphoribosyl transferase. You do not need to remember the enzyme name. This short form is enough, HGPRT. So the myeloma cells, now we started with two cells, one the B cell and the other is a myeloma cell. The myeloma cell, which is the cancer cell, is such that it lacks this enzyme. This enzyme HGPRT is not present in the myeloma cell. Since aminopterin blocks the de novo nucleotide synthesis pathway, myeloma cells cannot survive without a functional salvage pathway. This is the key point here. One of the components, which is aminopterin, is an inhibitor of the de novo synthesis of DNA. So aminopterin will now block one of the two major pathways of DNA synthesis, pushing the cell to survive on what is known as a salvage pathway. So when the de novo synthesis is stopped, the salvage pathway activates and it undergoes DNA synthesis. Now, for the salvage pathway to activate, because myeloma cells lack HGPRT, they die in HET medium. Now, for the salvage pathway to function, you need HGPRT. You need this enzyme, but the myeloma cells do not have this enzyme. So when you put this selection media, when you grow myeloma cells on this selection medium, these myeloma cells will die. But only fused myeloma cells, which have now acquired the property of functioning the salvage pathway, will survive. B lymphocytes have a functional salvage pathway, so they can survive in HET medium. So this is the key point. B lymphocytes can function through the salvage pathway, while myeloma cells cannot function through the salvage pathway. And both of these, when grown in this HET medium, only fused myeloma cells, which have both the properties of B lymphocyte and the myeloma, will survive in the HET medium. However, they have a limited lifespan and eventually die out since they cannot continuously divide. So B cell has another limitation. It is not cancerous. After a few divisions, B cell will die. So now we have a method to select for fused cells. Myeloma cells can progress indefinitely, but they cannot divide in the presence of HGPRT. B cells will divide for a few divisions and then die off. So selects for hybridomas, fused B cell plus myeloma, which inherit both the properties, the ability to survive using the salvage pathway from the B cell and the ability to divide indefinitely from the myeloma cell. So this is how an HET medium functions. These hybridomas survive in HET medium, ensuring only antibody producing immortal cells that remain in the medium to purify them. HET medium plays a crucial role in ensuring that only hybridomas are selected for further screening and mass production. So here is a schematic for you guys so that you will not get confused. So this is a immortal HAT sensitive plasmacytoma or a myeloma. Myeloma is immortal since it is cancerous, it can divide indefinitely, but it is HAT sensitive. And why sensitive? Because it has aminopterin, which will block the de novo pathway of nucleotide synthesis, while H and T, which are hypoxanthine, and T is thymidine. So this cell lacks the HGPRT enzyme, the hypoxanthine guanyl phosphoribosyl transferase. This enzyme is lacking, so it does not have the salvage pathway. So this is one kind of a cell, the immortal myeloma cell. This is dying in the presence of HAT. Now mortal splenic B cell. Splenic means those B cells that are isolated from the spleen. This also dies after a few rounds of divisions in the HAT. Now only the fused cell, which has both the properties of the immortal properties of the myeloma as well as ability to use the salvage pathway of nucleotide synthesis from the B cell, will survive. Immortality from the plasmacytoma and rescued ability to synthesize DNA due to restored thymidine kinase function. This is the function of HAT selection, in which we only screen for fused hybridoma producing our antibody of interest, not the B cells and not the myeloma cells. Now is it clear to the students? I have spent enough time on this topic for you guys to be clear about it. You understand what is the importance of HAT medium selection and how is it able to utilize the two different pathways of nucleotide synthesis for selecting the fused hybridomas. Is it clear to the students? That let me know in the chat. Very good. I hope it's clear for others as well. Okay, moving on to the next question. Gray's solution is used for removing dash. Gray's solution. Option A, PECs. Option B, B cells. Option C, myeloma cells. Option D, RBCs. What is Gray's solution and what is it used for? It is used for removing a certain kind of cells. Now which of these cells are removed when you apply Gray's solution? Is it option A, PECs, option B, B cells, option C, myeloma cells, option D, RBCs? Let us take a minute and collect some answers. For easier answer to this question, let me give you a hint. Gray's solution is used in the hybridoma technology. That is the hint. Now you can answer, what is it used for removing? Which kind of cells? Okay, let me check in the chat box what are the answers. Shravani has answered option D, Saij option D, and Shanmuga says I don't know. It's okay. It's okay. Uh, See, students, if you do not know the answer, or if you're guessing, if your answer is incorrect, you do not need to shy away. I will reveal what is the correct answer, and we will understand the concept. So it's a two-way street, right? If you know the answer, then well and good. Keep posting it in the chat box. I will always encourage that. But if you do not know, it's okay. Try doing guesswork as well. The correct answer to this question is option D, RBCs. So Saich and Shravani have answered correctly. Well done. Grace solution is used for removing RBCs in the first step of hybridoma technology, in which you isolate white blood cells. Sorry, you isolate the B cells from the spleen. That is when you use Grace solution to purify the isolated B cells of RBCs. Any kind of RBC will selectively die off. Now, what is the composition of Grace solution? It consists of these kind of. Grace solution is basically a hypotonic solution. It's a solution with low osmolarity. Ammonium chloride causes osmotic lysis of RBCs. Potassium bicarbonate maintains pH stability and helps in controlled lysis, and EDTA, which prevents clotting by chelating calcium ions. So it will prevent blood clot so that the cells can be isolated. Ammonium chloride will cause the lysis of RBCs, and potassium bicarbonate is a buffer for maintaining pH. Mechanism of RBC lysis: ammonium chloride present in the Grace solution enters RBC, causing an osmotic imbalance that leads to swelling and rupture. So in a hypotonic solution, ammonium chloride goes inside, increases the osmotic pressure inside the RBC, which leads to water flowing inside the RBC. Now the RBC will swell up and burst. White blood cells, including B lymphocytes, remain intact because they have a stronger membrane and regulator mechanisms to counter osmotic stress. So these regulator mechanisms as well as a stronger membrane is what makes RBCs different from, makes WBC different from RBC. Thus, only selectively the RBCs die off due to lysis or rupture. The remaining intact WBCs can be washed and used for further experiments. Example, hybridoma production. Procedure for RBC removal: collect blood or spleen cell suspension, ensure that cells are in sterile buffer, add Grace solution, mix the blood with 10 times the volume of Grace solution, and incubate at room temperature for 5 to 10 minutes. Then you centrifuge it, then you wash the cells to remove the supernatant and resuspend in fresh buffer. And then you resuspend WBCs in culture medium for further experiments. So this is the first step when you isolate the spleen from the mouse or the rat. Then how to isolate B cells from the blood from the spleen of the mouse? You use Grace solution to selectively kill RBCs, whereas preserving and isolating WBCs in a pure culture. And then this can be used for fusion with myeloma cells. Enrichment of B lymphocytes by cell sorting. So before cell sorting, this is treated with the Grace solution. Spleen is dispersed through nylon mesh. So there are all kinds of cells. There are monocytes, T cells, and basophils and B cells. We only want the lymphocytes here, which are B cells. So we kill off the RBCs using the hypotonic Grace solution. The ammonium chloride is the major player for lysis of the RBCs. So this is whole blood RBC lysis. Grace solution is commonly used hypotonic lysis buffer designed to selectively lyse red blood cells while preserving WBCs, including B lymphocytes used in hybridoma technology. So when you isolate the RBC, first you when you isolate the whole blood or the spleen, you incubate them with the RBC lysis buffer, the Grace solution. After lysis, it turns somewhat like this. After second round of lysis, and this is the final cell pellet in which you only get WBCs and you throw out the supernatant. Now, is it clear to the students what is Grace solution and which step of hybridoma technology it is used? It is used selectively for killing of RBCs and enrichment of B cells for the hybridoma technology. Is it clear to the students? Students, is it clear? Just let me know in the chat box. Then we'll move forward. Okay, great. Moving on to the next question. Question number 7. Aminopterin in HAT medium inhibits de novo synthesis of dash. Okay, we have discussed this at length. So this question should be a piece of cake for everyone. Aminopterin in HAT medium inhibits the de novo synthesis of dashed. Option A, nucleotides. Option B, lipids. Option C, amino acids. Option D, sugars. Aminopterin. So there are a series of follow-up questions on hybridoma technology because it is a very important concept which is still in use and in practice for research purposes, for immunotherapy, for any big drug development, for vaccine development. We need plasma cell to be immortalized using this hybridoma technology. This is a revolutionary technology. That is why I have many questions from the hybridoma technology, so that you get it by heart, you understand it totally, you understand the process, and you memorize the process. Aminopterin in HAT medium inhibits de novo synthesis of dashed. Let me quickly check the chat box. I will not wait for a longer time. Manjulata says option A. Swati is saying option A. And I know others would also be answering option A. Yeah, we have. Okay, Shanmugh says amino acids. Maybe let me reveal what is the correct answer to this question. Correct answer to this question is option A, nucleotides. I think Shanmugha was not clear in the HAT explanation in the previous question. Aminopterin is an inhibitor for the de novo pathway for nucleotide synthesis. So let us understand what are the two pathways for nucleotide synthesis. There is a de novo pathway for nucleotide synthesis. Small metabolites like sugars and amino acids present in the media. This is converted into a nucleotide, and when I say a nucleotide, it has the pentose sugar, the nitrogenous base connected via the N-glycosidic bond, the phosphate group to the 5th carbon of the pentose sugar, and utilizing the small metabolites like sugars and amino acid present in the media, de novo pathway synthesizes this. There is another pathway called the salvage pathway, where the salvage pathway utilizes parts of degraded nucleotides to salvage them and make a new nucleotide. So aminopterin blocks the dihydrofolate reductase enzyme, which is a key enzyme in the de novo pathway. So aminopterin affects the production of nucleotides. No new nucleotides are formed. So therefore, the cells have only to rely on the salvage pathway. Now in myeloma cells, where the salvage pathway is also sabotaged, only the hybridomas can survive when they have the properties of both the plasma cell as well as the myeloma cell. Because these myeloma cells do not have HGPRT, which is a hypoxanthine phosphoribosyl transferase converting hypoxanthine and thymidine from HAD media to new nucleotides. So it does not have this enzyme, these myeloma cells, so they do not survive on HAT media, while B cells can survive on HAT media. So only those cells which are fused with the B cell and the myeloma cell have the property to utilize the salvage pathway for new nucleotide synthesis. Pure B cells will die off after a few rounds of replication since they are not immortalized, and pure myeloma cells will die off since they do not have both the salvage pathway and the de novo pathway because of aminopterin present in the media. Now is it clear to everyone that aminopterin is a blocker of de novo nucleotide synthesis pathway? I hope it's clear to everyone. guys, let me know in the chat box that is it clear to everyone. So Shanmuga, amino acids is not the correct answer. The correct answer would be nucleotides. Aminopterin is in the de novo synthesis of nucleotides.
Sir, thank you, sir.
Okay, great. And this is the same thing we have discussed over and over. Aminopterin is a folic acid antagonist that inhibits de novo synthesis. It blocks this enzyme dihydrofolate reductase, preventing cells from synthesizing purines and pyrimidines via the de novo pathway. As a result, only the cells that can use the salvage pathway, such as hybridomas, which inherit this ability from the B cells, can survive in HET medium. Okay, let's not stall any more time in this question and move on to the next question. Question number 8. Pinocytosis is the process of internalizing dash. Pinocytosis. Option A, bacteria. Option B, virus. Option C, antibodies. Option D, fluids. Now, what do you know about pinocytosis? It is an internalization process, but what is the substance that is getting internalized in pinocytosis? Is it bacteria? Is it a virus? Are these antibodies or are these fluids? Let's wait for some time and quickly collect some answers. I would encourage all the participants to at least guess the answer, what you think is the correct answer. Then we move on quickly through the concepts. Okay, let me quickly check the answers in the chat box. Okay, Swati has answered option D. Shanmuga says fluids. Manjulata option D. Shravani also says option D. You guys are absolutely correct. Option D is the correct answer. Pinocytosis is the process of internalizing fluids by the cell. So these are a brief stepwise overview of pinocytosis. There are solutes present in the fluid, which is the extracellular fluid. Solutes bind to the cell membrane. Cell membrane starts to fold inwards in the extracellular fluid. Cell membrane continues to fold. Both ends of the fold meet, producing a pinosome. So there are such small structures called a pinosome, in which solutes means substances which are dissolved in fluid is taken inside, and then the cell membrane pinches off inside the cell, forming a double membrane organelle which shares a similar characteristic of the plasma membrane as a cell, known as the pinosome. So this is pinocytosis in summary. Pinocytosis is a process by which cells take in dissolved substances and fluids from outside the cell. It is also known as fluid endocytosis or cell drinking. In slang terms, it is also called cell drinking. Cell is drinking something. How does it happen? The cell membrane folds inward, creating small pockets that trap fluid and dissolved substances, like this. Pinocytosis, you see? These are the solutes, small substances. The cell membrane is folding its membrane. Cell membrane closes around the pockets, forming vesicles known as pinosomes. The vesicles fuse with lysosomes, where digestive enzymes break down the molecules. And why is pinocytosis important? Pinocytosis is a way for cells to take in nutrients like vitamins, fat droplets, and antigens. It is also involved in transporting toxic ions from the apoplast to the vacuoles of the shoot in the salt accumulating halophytes. So, pinocytosis is one of the major ways in which antigen is engulfed inside the cell, processed, and then presented in antigen presenting cells. So there are many other modes of uh, cell engulfing, which is phagocytosis of solid particles, pinocytosis for fluids, and receptor mediated endocytosis. I hope it is clear to everyone. You guys have answered it very correctly, so I do not have any doubt in that. But just let me know in the chat box whether it is clear to everyone. We have more questions to cover, so let's pace up a bit, but not on the explanation part. I will go as slow as possible for all of you guys to understand. Okay, thanks for letting me know. Moving on to the next question. Question number 9. Permeabilization of cells is performed routinely with dashed. Permeabilization of cells is performed routinely with option A, glycerol, option B, glutaraldehyde, option C, Triton X100, option D, paraformaldehyde. Cell permeabilization. Which of these is used as a cell permeabilizing agent? Routinely, very commonly. Let's wait for some time and collect some answers in the chat box. Cell permeabilization. If you think of the property, then you will know of these answers. Like what is the property of the chemical or reagent that are present in the options? So then you would easily reach the correct answer. Okay, time for me to quickly check the chat box for answers. Manjulata says option C. Swati is also saying option C, and Shanmuga is saying option C. Someone has raised a hand. Kati has raised a hand, is it? Kashiram. Yeah, Kashi, you have something to say, or maybe you can post it in your chat box. Kashiram also, okay, he was answering option C. Shravani says option D. What is the correct answer to this question? Option C, Triton X100 is the correct answer. Well done, Manjulata, Swati, Shanmuga, and Kashiram. You guys have answered correct. Shravani, you missed by a bit. It's not paraformaldehyde, but it's Triton X100. Now let us understand, let us see what is Triton X100. Triton X100 is a non-ionic surfactant. And by surfactant, in common terms, I mean detergent. It's a detergent. Triton X is a non-ionic detergent with a hydrophilic. So this part is the hydrophilic segment with OH groups, polyethylene oxide chain, and a hydrophobic aromatic carbon group. This is the hydrophobic segment. Hydrophilic segment and a hydrophobic segment. So Triton X100 molecule is a detergent with a hydrophilic part and a hydrophobic part. Now how does it aids in cell permeabilization, and where is it used? Uses of Triton X in research and industry: cell lysis and membrane permeabilization, used in Western blotting, immunocytochemistry, ELISA to permeabilize cells and allow antibodies to enter. Solubilizes membrane proteins without denaturing them. So these kind of representation you see is a hydrophilic head group, which is the hydrophilic head that we saw here, hydrophilic segment, and then the hydrophobic tail group, which is mimicking a lipid structure, a single monolipid structure in the bilayer, and this can form either micelles with a polar and non-polar head group. It can aggregate proteins without denaturing them. Triton X is also used in protein solubilization, in DNA RNA extraction, and viral and bacterial inactivation, because It disrupts the lipid membrane of viruses and bacteria. While in industrial application, it is present in cleansing agents, emulsifiers, and some cosmetics. So Triton X100 is a detergent or a surfactant used for cell membrane disruption and permeabilization because it forms micelles, it can aggregate proteins without denaturing them, and it is useful as a detergent forming two interfaces, hydrophobic and hydrophilic interfaces, perfectly. Whereas paraformaldehyde is used for cross-linking or fixing, killing cells, preserving or fixing the cells in their pristine state, cross-linking the cells in the state they are. So the correct answer to this question is Triton X100, which is a surfactant or a detergent. Is it clear to the students? Just let me know in the chat box, then we can move ahead. Hello? Okay, I think my screen froze for a minute. Shanmuga is asking, sir, can you show the question again? Okay. The question was this: permeabilization of cells is performed routinely with dashed glycerol, glutaraldehyde, Triton X100, paraformaldehyde. Cell permeabilization is, of course, done by a detergent, which is Triton X100. Okay, is it clear? Can we move forward? Okay, we have a few more questions today, so we will move forward. Question number 10. Very good, thanks for the confirmation. Question number 10. The side scatter channel SSC detects dash. So now we have come to a separate paradigm of the lectures. The side scatter channel detects dash. Option A, cell size. Option B, cell granularity. Option C, cell membrane permeability. Option D, cell shape. The side scatter channel. And when I say side scatter channel, I am referring here to the FACS or MAX sorting. What is the side scatter channel plot detecting? Is it detecting cell size, cell granularity, cell membrane permeability, or cell shape? Let us wait for a minute and collect some answers. Then I'll reveal what is the correct answer to this question. Let me check in the chat box for answers. Okay, Manjulata is saying I don't know. I think many of us present here do not know the answer to this question. The side scatter channel here detects option B is the correct answer, cell granularity. I think someone has answered. Shravani, answer B. Yes, Shravani, you are correct. Side scatter channel detects cell granularity. I hope you guys are giving these answers on your own and not just googling in a minute or so. Answer B is the correct answer. Cell granularity is detected by the side scatter channel. Now, what are these different scatters that we see when a cell is passed through a flow cytometry or detected in a flow cytometer? So, suppose this is a cell. Inside the cell there is a nucleus and there are granules present here. Now there are two kinds of parameters that a flow cytometer measures. One is the forward scatter and the other is a side scatter. In flow cytometry, side scatter is a parameter that provides information about cell's internal complexity. See, these granules will scatter these rays, incident light source, granules that are present inside the cell. So, side scatter is a measure of cell's internal state or complexity. It measures how a light is scattered at a 90-degree angle when a laser beam hits the cell. And what is forward scatter? Forward scatter, on the other hand, will detect this size. When you shine a ray of light through a cell, now the side scatter will tell us how granular a cell is, while the forward scatter will tell us how big the cell is. Like these lines you see, the forward scatter measuring the cell size, while side scatter measuring cell complexity. So a higher side scatter indicates more granules or internal structures. Granulocytes like neutrophils have higher SSC due to cytoplasmic granules. Monocytes have moderate SSC, fewer granules, and lymphocytes have low SSC, small or smooth cytoplasm. Low SSC is less granularity. Red blood cells and platelets have very low SSC because they lack internal structures. Red blood cell does not even have a nucleus. And this is how you read a flow cytometry plot or a FACS plot. Increasing side scatter in the y-axis and increasing forward scatter in the x-axis. So as further you move in the y-axis, there is increasing side scatter which depicts granular cells. Like this population, granular cells, and you increase in the x-axis, bigger cells. So here is the eosinophils or basophils. Monocytes will appear somewhere here with medium side scatter but large size, and the lymphocytes which do not have much side scatter because they are agranular but with a moderate size. So this is how you read a x and y axis of a FACS plot or a flow cytometry plot. With side scatter on the y axis, increasing side scatter depicting granularity of the cell, and increasing forward scatter depicting increasing size of the cell. I hope it's clear to the students. Just let me know in the chat box and we will move ahead. Did everyone understand the side scatter in a FACS plot? Okay, let's move forward. Question number 11. Cytochrome C is released during apoptosis from dashed. Option A, Golgi bodies. Option B, nucleus. Option C, mitochondria. Option D, endoplasmic reticulum. Cytochrome C is released during apoptosis from which of these cellular structures? Is it Golgi bodies? Is it nucleus? Is it mitochondria or endoplasmic reticulum? Let me know of your answers in the chat box. I will wait for a minute or so. Okay, let me check the chat box for quick answers. Manjulata is answering option C. Shanmuga says, I think B. Shravani, option C. Kashiram, B. Mitochondria. I think C is mitochondria, but I get your answer. Network problem. No issues. No issues, Kashiram. You're trying to follow up. I'm happy with that. Please follow up. You can watch these recorded videos as well, and these slides will be shared with you. So you stay with me, stay together. We have few more questions and the class will end soon. And the correct answer is option C, mitochondria. Very well done. Kashiram, Manjulata, Shravani have all answered correct. And Shanmuga, it is not the nucleus, but cytochrome C is will be released from the mitochondria, because cytochrome C, if you remember, is an integral part in the electron transport system, which is present in the mitochondrial membranes and not in the nucleus. Now, if you look at the mitochondrion, cytochrome C, these molecules, these brown molecules, are present in the membranes of the mitochondria, the inner membrane of the mitochondria, for the electron transport chain, and it is necessary for signaling the cell into survival or into apoptosis. Now, if the cytochrome C is oxidized or is exposed out of the mitochondria by the enzyme COX or cytochrome C oxidase, then it signals apoptosis, formation of apoptosome by recruiting proteins called APAF1 and procaspase 9, which are the proteins of the apoptotic pathway. When the same cytochrome C is reduced and present inside the mitochondria by cytochrome C reductants like superoxide, ascorbate, glutathione synthase, this leads to cell survival. So cytochrome C exposure from mitochondria into the cell signals apoptosis, while presence of cytochrome inside the mitochondria in the electron transport chain signals survival. Now, what is the normal condition or function of the cytochrome? Cytochrome C is localized in the mitochondrial intermembrane space. It participates in electron transfer from complex 3 to complex 4 in the ETC, aiding in ATP production. Apoptosis, activation, and mitochondrial outer membrane permeabilization. What happens in apoptosis? A phenomenon happens which is called MOMP or MOMP, mitochondrial outer membrane permeabilization. The outer membrane of mitochondria gets permeabilized. Cellular stress triggers pro-apoptotic proteins backs and back, leading to MOMP or mitochondrial outer membrane permeabilization. MOMP causes cytochrome C release into the cytoplasm, marking a point of no return in apoptosis. So once there is MOMP and cytochrome C is exposed outside in the cytoplasm, there is no return for the cell. It is destined for apoptosis. And then there is formation of apoptosome by caspase activation. So there is MOMP, which is leading to apoptosis, and cytochrome C, SMAC, these are all indicators of apoptosis which get exposed in the cytoplasm. And whenever there is cellular stress, BS3 only proteins, they activate these pores or help in MOMP, which is membrane permeabilization, using these proteins. So cytochrome C is exposed in the mitochondria into the cytoplasm, which is a signal for apoptosis. And normally it is part of the electron transport system, aiding in ATP production in electron transfer from complex 3 to complex 4. Is it clear to the students? Very good. Thank you. Moving on to the 12th question. Calcium sequestration mainly is performed by dashed. Calcium sequestration is mainly performed by which organelle of the cell? Option A, Golgi body. Option B, lysosomes. Option C, endolysosomes. Option D, endoplasmic reticulum. Calcium sequestration. You might have heard a particular organelle which is associated with calcium all the time. That is the correct answer to this question. I have given you a big hint. Calcium sequestration mainly is performed by which of these cellular organelles? Let us wait for a minute and collect some answers. Okay, let me quickly check what are the answers in the chat box. I see only one answer from Kashiram. Endoplasmic reticulum. Okay. What about others? Do you know the answer to this question? Do you want to answer? Or this is too much for today? We have already had 12 questions. Bear with me a little longer. 3 more questions to go. Question number 15 will be the last question, I promise to you. Okay, let me reveal what is the correct answer to this question. Option D, endoplasmic reticulum. And Kashiram, congratulations, you have answered very correctly. Calcium sequestration is mainly performed by the endoplasmic reticulum in the cell. Here is a structure for all the complexes, all the organelles involving calcium. Now there are channels on the outer membrane, plasma membrane of the cells, which are called TRPC channels. There are many other pumps which aid in transport of calcium. There are mitochondria. Mitochondria also utilizes calcium via these channels, VDAC channels, MCU complex. Lysosome also requires calcium for many of its function, and the enzymes require calcium as a cofactor. But the major reservoir of calcium here is in endoplasmic reticulum through this protein SERCA and other transporters of calcium which are present on the membrane of the endoplasmic reticulum. The ER. Okay, there is a chat. Can you show the question again? Shravani is asking. Okay, I'll just show the question again. Calcium sequestration mainly is performed by which of these organelles? The Golgi body, the lysosomes, endolysosomes, or the endoplasmic reticulum? This was the question, and the answer is the endoplasmic reticulum. And we were understanding how calcium is stored in the ER. The ER is a major calcium storage organelle maintaining high calcium concentration inside. Release and uptake of calcium are tightly regulated for cell signaling, metabolism, and apoptosis. So many such signaling, many GPCR signaling involves release of calcium inside the ER into the cytosol as a result of activated G protein signaling. Right? If you remember G protein signalings, calcium release from the ER, inositol 145 trisphosphate receptor, INS 145P3R, which is this. This receptor releases calcium into the cytoplasm when activated by IP3, important in cell signaling and apoptosis function. So this receptor is activated by the G signaling, GPCR signaling. Ryanodine receptor, RYR, another ERCA2 plus calcium release channel, especially important in muscle contraction and neuronal signaling. So calcium also plays a major role in muscle contraction, in contraction of myosin and troponin. Calcium uptake into the ER, sarcoendoplasmic reticulum or SERCA channels, Ca2 plus ATPase pumps calcium back into the ER using ATP. So they use energy and pump the cytoplasmic calcium back into the ER, maintains calcium homeostasis and prepares the ER for future signaling. So this is the SERCA, this is the inositol IP3 receptor, this is the RYR receptor present in the muscles and neurons, IP3 receptor for signaling related calcium reservoirs, and the SERCA which is utilizing an ATP and pumping in back the calcium to the ER. Maintains calcium, store operated calcium entry, SOCE and replenishment. Whenever ER calcium level drops, SOCE activates to refill stores. and STIM1 detects low calcium and activates ORI channels at the plasma membrane to bring extracellular calcium. So these are names of certain proteins and channels present on the surface of ER, which helps in maintaining the homeostasis of calcium concentration inside the ER as well as outside in the cytoplasm, and a shuttling between these two concentrations necessary for cell functions like signaling events, calcium as a second messenger, and contraction events in the muscle, and synapse firing in the neurons. This is the SOCE, which brings in more calcium to replenish the pools of calcium in the cytoplasm. Is it clear to the students that calcium storage is majorly happening in the endoplasmic reticulum via these channel proteins? If it's clear, then we'll move forward. Just let me know in the chat box. Or if you want to look at this diagram again. Okay, thank you. Moving forward to the 13th question. In Golgi bodies, proteins are covalently linked with sugar in which part of the Golgi? In the whole organelle Golgi bodies, proteins are covalently linked with sugar in dash. A. Cis phase. Option B. Medial Golgi. Option C. Trans phase. Option D. Ribosomes attached to Golgi. In which of these compartments in the Golgi do you think proteins are covalently linked with sugar? Let me know your answers in the chat box. I will wait for a minute and we'll quickly check the chat box and reveal what is the correct answer to this question. Okay, it's time for me to check the chat box and see what are the answers. Kashiram says B. Prashanti says D. Shanmuga has changed his option. Sir, trans phase. Okay, Shanmuga is saying option C. Okay, let me reveal what is the correct answer to this question. Correct answer to this question is option B, medial Golgi. So only Kashiram has answered correctly. Prashanti and Shanmuga, better luck next time. But the option B is the correct answer, medial Golgi. So let us understand the different compartments in the Golgi apparatus. So a Golgi apparatus has three compartments: the cis compartment, medial compartment, and the trans compartment. The cis compartment is the closest to the endoplasmic reticulum. Then in the middle we have the medial compartment, and towards the plasma membrane is the trans compartment. Now what happens in the cis compartment? Mostly phosphorylation of lysosomal oligosaccharides and removal of mannose, O-linked glycosylation. The Golgi apparatus is responsible for protein glycosylation, which means attaching sugar molecules to proteins, which was the question of interest. This process occurs mainly in the medial Golgi and trans Golgi compartments. Cis phase receives proteins from the ER. Medial Golgi modifies proteins, including adding sugar residues like glycosylation and the transphase sorts and packages proteins for transport. So transphase does. So Shanmuga was right. The transphase does also include glycosylation of proteins, but most of the glycosylation happens in the medial Golgi, while transphase is majorly associated with sorting and packaging of proteins for transport. Cis for receiving, glycosylation and modification in the medial, and export or transport in the transphase. Ribosomes are not attached to the Golgi, they are found in the rough endoplasmic reticulum, so ribosome cannot be an option. So option D is incorrect. But majorly the glycosylation or addition of sugar residues, carbohydrate residues to the proteins happens in the medial Golgi, while in the trans Golgi, sorting of these molecules and cis receives the protein molecules from the ER. Is it clear to the students? Cis receives the protein, medial attaches the sugar group, and trans transports the protein. 3 parts of the Golgi. Is everyone clear? Very good, very good. Coming to the 14th question. Just one more question left to end this session. We are in time. 14th question. Glycosylation is again an easy question. Glycosylation is dashed. Option A, addition of amino acid to a growing peptide. Option B, removal of a sugar from a protein. Option C, addition of a sugar to a protein. Option D, addition of ubiquitin to a protein. Come on guys, answer this very fast so that I can check the answers, reveal the correct answer, and move on to the final question of this session. What is glycosylation? What does it mean? What kind of modification is done? Sometimes these questions can be very confusing, but you have to read in between the lines and look at all the options very carefully. They are very similar looking options. Kashiram says, sorry sir, I don't know, please explain it. Okay, I will explain it. We will go through the explanation. Any more answers? What is glycosylation? Shanmuga is saying option C. Shravani says B. Okay. And I think that's it. Now let me reveal what is the correct answer to this question. Correct answer is addition of a sugar to a protein. We just encountered this in the previous question. Glycosylation is nothing but addition of a sugar moiety to a protein. Yes, glucose means sugar, Kashiram. Not just glucose. There are many varieties of sugars. There can be glucose, galactose, and modifications with acetyl groups, GLC, NAC, and modified glycans added to a protein. So glycosylation is a post-translational modification. After a protein is translated, is a post-translational modification where sugar molecules like carbohydrates are covalently attached to proteins. It plays a crucial role in protein folding, stability, and cell signaling. Now there are many types of glycosylation. N-linked glycosylation. Sugar is attached to the amide nitrogen of asparagine. O-linked glycosylation. Sugar is attached to the hydroxyl group of serine and threonine. And why the other options are incorrect? Adding of amino acid to a growing peptide is translation itself, because translation happens in ribosome. Adding of a amino acid to a growing peptide. This is not glycosylation. Removal of a sugar from a protein refers to deglycosylation. Like you remove D-glycosyl. You remove the glycosyl group, the carbohydrate group from the protein. Addition of ubiquitin is called ubiquitination, which is another important post-translation modification for protein degradation. Ubiquitination targets the target protein into proteasomal degradation. Is it clear to the students? Like what is glycosylation? Is it clear to the students? Let me know in the chat. Very good. Quickly moving on. Okay, so here is a representation here showing O glycans and N glycans and their differences. N glycans, asparagine glycans, O glycans, oxygen of the serine and threonine, the amino acids with oxygen. Okay, now a related question, the last question of this session, question number 15. Most common type of glycosylation is option A, N glycosylation, option B, O glycosylation, option C, C-linked glycosylation, and option D, phosphoglycosylation. What is the most common type of glycosylation which you find in proteins? Let me wait for a minute and collect some answers in the chat box. Most common type of glycosylation. Okay, I already see some answers in the chat. Shanmuga says option A, Kashiram option A, Prashanti option A, Faru is option also saying option A, and Shravani option A. And you guys are all correct. Congratulations. N glycosylation is the correct answer. N glycosylation is the most common type of glycosylation found in proteins. And just as we saw, N glycosylation happens both in the ER and the Golgi, while O glycosylation mainly happens in the Golgi apparatus. Now what is N-linked glycosylation? Is the most common type of glycosylation. It involves attachment of a sugar molecule to the amide nitrogen of the asparagine in the consensus sequence, asparagine X serine threonine. So because it is attached to the amide group or the nitrogen group, that is why it is named N-glycosylation. O-linked glycosylation attached to the oxygen group of the serine threonine. And in this sequence, asparagine X is any amino acid except proline and serine threonine. In these triple sequences, the asparagine amino group will be attached with the glycosyl group, which is the sugar group. And why is this important? It occurs co-translationally in the endoplasmic reticulum and continues in the Golgi apparatus. It helps in protein folding, stability, and cell recognition. It is an essential for the function of many membrane and secreted proteins. So why does this N-glycosylation, where does this happen? It happens in the ER, which in the rough endoplasmic reticulum harbors the ribosome, where co-translationally, once the nascent polypeptide is translated, side by side it is also undergoing glycosylation in the ER. And then the peptide, once glycosylated, follows to the Golgi and then undergoes more glycosylation. And this glycosylation helps in proper folding of the protein, stability of the protein, and cell recognition. And this is essential for many membrane and secreted proteins. So which you guys have answered correctly. The answer to this question is N-glycosylation, which is the most common type of glycosylation. Is it clear to the students? N-glycosylation happens in both the ER and the Golgi apparatus, while the O-glycosylation only happens in the Golgi apparatus. And here are some examples. Mannose group added in the N-linked glycosylation, hybrid groups which includes mannose, glucose, and galactose, right? So with this, we have come to the end of our problem solving session. I will stop sharing my slides and turn on my video chat. Yes, I will stop presenting. Yeah. So with this, we have come to the end of our session, and I hope in time, because it's already 5:02 p.m. So as mentioned earlier in the start of the class, this is a recorded session. The video will be uploaded in the YouTube. All these slides with all the information will be uploaded in the Google Drive, which will be provided, both the YouTube link and the Google Drive link will be provided to you by the NPTEL authorities, which you can access later. And do read your, do go through your lecture series, do attend your live sessions. So there are multiple TAs which are taking this live session. You can attend any of the ones which you like and which fall in your timing. And yeah, see you soon next week with problem week set 8. And thank you for joining. I hope you enjoyed this class. I will end this call right now. Thank you.